Hello, my name is Joshua Mugabi. I welcome you to Biology King. In this session, we are going to look at membrane structure. Outline the two key qualities of plasma membrane. 1. Semi-permeability. Something can cross the membrane, other things cannot cross. Secondly, it is selectivity. Cells can control the passage of certain materials across the membrane. Distinguish between passive transport and active transport. Let's start with the passive. Movement from a region of higher concentration to a region of lower concentration along the gradient. That is passive transport. Active. This is the movement from a region of low concentration to a region of higher concentration. And it is always against the concentration gradient. Passive does not require the expenditure of ATP, that is energy. For example, simple diffusion, facilitated diffusion, and osmosis. For active, it requires the expenditure of ATP or energy. Also, it requires membrane proteins or the protein pumps. Define simple diffusion. This is the net movement of particles from a region of high concentration to a region of low concentration until when the equilibrium is reached. That is simple diffusion. Describe the process of facilitated diffusion. Now, facilitated diffusion is a passive transport mechanism for materials that cannot freely cross the membrane. It is facilitated and it is passive. Large and charged substances cannot freely cross the hydrophobic layers. Those are the ions and macromolecules. Therefore, the membrane proteins may include the channel protein, which forms a hydrophilic pore, and they may also include carrier proteins, which translocate substances. List 3 molecules that are transported by simple diffusion and facilitated diffusion. Simple diffusion, we have materials like oxygen, carbon dioxide, lipophilic substances like steroids. For facilitated diffusion, we have substances like ions. For example, we have sodium, potassium. We have macromolecules like glucose, starch, and others. Describe how the structure of the potassium channel facilitates the diffusion in nerve cells. integral proteins with a hydrophilic inner pore via which potassium ions may be transported. The inner pore contains a selectivity filter in order to restrict the passage of alternative ions. Channels adapt an open stroke closed conformation based on 1. Membrane polarity, that is voltage gated. Secondly, binding of a neurotransmitter, which is the ligand-gated. Describe the relationship between solutes and solvents in a solution. A solute is a substance that is dissolved in a solvent. Due to its polarity, water is often termed the universal solvent, but won't dissolve non-polar molecules. Define osmosis. The net movement of free water molecules across a semi-permeable membrane from a region of low solute concentration to a region of high solute concentration until equilibrium is reached. That is osmosis. Differentiate between hypertonic, hypotonic, and isotonic solution. Now, hypertonic solute concentration, it has a higher solute concentration relatively. And what is the effect on the cell? It will cause water to flow out of the cell into the hypertonic. Hypotonic solution. The concentration of the solute is lower relatively, or it has a lower solute concentration. And its effect on the cell, it is going to cause the water to flow into the cell from the hypotonic solution. Then we have the isotonic. The solute concentration equal solute concentration relatively on both sides. And what is its effect on the cell? There will be no net water movement. Define osmolarity. Osmolarity is a measure of solute concentration as defined by osmol per liter. Distinguish between primary active transport, or what we call direct, and secondary active transport, what we refer to as indirect. Let's start with direct. ATP hydrolysis is used to mediate transport by causing a conformation change in the protein pump, thereby translocating the membrane molecule against the gradient. That one is primary active transport, which we call direct. The secondary active transport, indirect transport, is coupled to another molecule moving along an electrochemical gradient. So it leads to co-transport. That one is what we call secondary active transport or indirect. Describe how the structure of the sodium-potassium pump enables active transport in nerve cells. Sodium ions are translocated and released from the cell, thus exposing extracellular binding sites to potassium. For potassium, now 2 potassium ions are going to be attached to these sites, and the pump is deforcibly. Now 3 sodium ions attach to the intracellular binding sites on the protein pump, and the ATP is hydrolyzed, phosphorylated the pump, and changes its conformation. Now, this returns the pump to its original conformation, internalizing the potassium ions. Outline the role of vesicles in the transport of materials between organelles. Polypeptides destined for secretion are synthesized by the ribosomes on the rough endoplasmic reticulum. They are transported to the Golgi body via vesicles. These ones are formed from the endoplasmic membrane. The Golgi body will potentially sort, store, and modify these secretory products. The proteins are then transported by another vesicle to the cell membrane for secretion, what we call exocytosis. External materials can be internalized into cellular vesicle via endocytosis for digestion by lysosome. Use the diagram below to explain the process of endocytosis. Endocytosis is the process by which large substances enter the cell without crossing the membrane. That is endocytosis. An invagination of the membrane forms a flask-like expression which envelopes the extracellular material. The invagination is then sealed off to form an intracellular vesicle containing the material. Differentiate between phagocytosis and pinocytosis. Phagocytosis, the process by which solid substances are ingested. Pinocytosis is the process by which liquids or solutions are ingested by the cell. Use the diagram below to explain the process of exocytosis. Exocytosis is the process by which large substances exit the cell without crossing the membrane. Vessels fuse with the membrane of the plasma, expelling their contents into the extracellular environment. Exocytosis adds vesicular phospholipids to the cell membrane, replacing those lost via endocytosis. Hello, my friends. Thank you very much for participating in Biology King. I encourage you to subscribe.